Good day, this is the Professor, CBMD PhD, and today is Wednesday, September 27, 2023. It is 5:46 and 8 seconds a.m. Eastern Standard Time, and today's research, mutation and cancer. So let's come to it here. Mutation-specific peptide vaccine against midline gliomas used in patients, date September 21st. 2023. Source: German Cancer Research. As tumor vaccines can help the body fight cancer, these vaccines alert the patient's immune system to proteins that are carrying cancer, typical alterations. The physicians and cancer researchers have now treated adult patients with advanced midline gliomas, difficult to treat brain tumors, with a peptide vaccine for the first time. The vaccine mimicked a mutational change in a histone protein, typical of this type of cancer. The vaccine proved to be safe and induced the desired immune responses directed against the brain tumor. As crucial for the success of cancer vaccinations are protein structures by which the immune cells can recognize the cancer and which are not present or only to a small extent on healthy cells. Mutations in the tumor genome often lead to protein structures that are altered in a way typical of cancer. Diffuse midline gliomas are among the most aggressive brain tumors. They usually occur in children and young adults near the brainstem and are therefore difficult to access surgically. Chemotherapy or radiation therapy also have limited effectiveness. In this type of cancer, mutations characteristically occur in the gene encoding histone H3, H3K27M, a packaging protein of DNA. The mutation gives rise to a protein structure, a so-called neoepitope, that can be recognized as foreign by the patient's immune system. And such mutations, which occur in identical form in many patients, are rare in cancer. They literally lend themselves to the development of tumor vaccines because they occur in all cancer cells. Since the mutated histone is causative for the development of midline gliomas, this means that vaccination against the mutated protein gets to the root of the problem. The scientific investigators and researchers synthetically restructured the section of the histone H3 protein with the characteristic mutation. Using this peptide, they were able to curb the growth of the H3K27M mutated tumors in a mouse model. In parallel, the researchers and physicians treated 8 adult patients with a peptide vaccine in time-limited, individual, curative trials. As these patients who could not be enrolled in the trial protocol suffered from diffuse midline gliomas with H3K27M mutation that progressed after standard therapy, some of the affected individuals received therapy with immune checkpoint inhibitors in addition to tumor vaccination. No serious side effects were observed in any of the vaccinated patients. 5 of the 8 treated patients developed specific immune responses against the mutant protein, which were dominated by CD4 T helper cells, and one of the patients who had shown a strong immune response. The tumor regressed completely and she remained tumor-free for 31 months. The vaccine peptide, which is comparatively long at 27 amino acids, worked in patients with different HLA variants. HLA proteins are responsible for the presentation of the mutant peptide on the cell surface and differ from person to person depending on their genetic background. The scientific investigators and researchers also observed that immune responses decreased over time, so repeated administration of the vaccine could support a sustained effect. The researchers cannot make any further statements about the efficacy of the vaccination based on these treatments. In any case, the current study has given them valuable information that will help them to further optimize the development of brain tumor vaccines in the future. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.